All right, well, good afternoon, everybody. My name is Alejandro Hernandez Rosas, as was just mentioned, and I am the PhD candidate in the Florentarius laboratory, and was to explain the person, looking at these roots and digging up trees and all this other stuff. And welcome to be here, and hopefully do a good job at representing what we do in the Florentarius laboratory. As the name of the title states, I'll be going over the management of Phytophthora crown and root rot in pistachio. Right, so just to kind of give a brief overview, Phytophthora species are not true fungal pathogens. These are organisms of their own. They're called oomycetes, commonly known as water molds. They're fungal-like, but something completely different. These are soil-borne pathogens that are favored by standing water, and the reason is because they actually have these unique spores that are called zoospores that actually give them the ability to swim through water and then cause infection. Right, so when we think of more generalities in terms of Phytophthora diseases, they're known to exist in canal and river water. So these surface water irrigation sources, we know that these swimming spores are present, and most likely how they are introduced into orchards. And to date, we haven't seen Phytophthora in well water. That hasn't been detected yet, so that is the good news. But other factors that kind of influence Phytophthora diseases are going to be things like poor water infiltration that causes this standing water in a particular orchard. And just to kind of give a quick overview in terms of the disease cycle, right? These Phytophthora species persist in infected root tissue or crown tissue under the right conditions. They form these resting spores, these oospores, which survive in the soil or on the root tissue. And then during favorable conditions like flooding or over-irrigation, you have these formation of sporangia and release of zoospores. And these zoospores are really what causes this explosion in Phytophthora crown and root rot disease. And some of the classic symptoms of Phytophthora crown rot in pistachio is going to be this excess of gumming seen at the base of the pistachio tree. Right, you kind of see this tannish to yellow clear gumballs. If you were to take a knife or a hatchet and just kind of look at the bark underneath, you'll see this necrotic tissue beginning to girdle that entire tree trunk. This girdling eventually causes the quick decline of the pistachio tree entirely, and these are the kind of classic field diagnostic symptoms that we use to quantify the disease. So Phytophthora manifests in two ways, the crown rot phase and the root rot. Primarily what we see with pistachio is this crown rot phase, and what this slide seeks to kind of emphasize is that these infections truly initiate at that soil line. They're not infecting the root and making their way up, but instead what we actually see is an infection starting at that soil line where you're going to be having that water pooling up right at the top of the soil. And other characteristic symptoms of Phytophthora crown rot infections is going to be these concentric rings seen at the margin of infections. And then, as I mentioned, they do girdle the entire tree trunk, and I don't know how easy you could see it, but you see this kind of black necrosis around the tree trunk, and really it's cutting off the water uptake, and that's what kind of causes this quick decline in infected pistachio trees. And once those infections kind of go into the scion, you see the formation of these larger black gumballs that form on the scion portion if infections go that far up. And I do want to emphasize that we have seen Phytophthora crown rot infections affecting trees at all life stages, from newly planted trees that haven't even been grafted yet to 1 to 2 year old grafts, and then much older trees as well. So this is a huge issue that we continuously get several farm calls and samples submitted every year, regardless of that tree age. And then, as I mentioned, the other phase of what this disease can manifest is in the root rot symptoms. And right, this root rot phase is going to be attacking those feeder roots. And you can kind of see some of those photos on the far Far left, you're right, where you kind of see this black necrotic root tips, and you kind of see this overall decrease in the root mass. And infections for the root rot aren't going to be causing a quick decline like what we see with crown rot, but you rather see this kind of gradual decline of the pistachio tree as a whole. And funding from the California Pistachio Research Board allowed the Trias lab to do these surveys and assess the situation of Phytophthora diseases in pistachios. What we found was that there's 3 Phytophoras that are responsible for this disease. That's going to be Phytophthora niederhauseri, Phytophthora mediterranea, and Phytophthora taxon walnut. Out of those 3 species, we do see niederhauseri a lot more prevalent throughout the main pistachio growing regions. And I do want to emphasize that these are new species that have been described affecting pistachio, and part of why we think they're beginning to be such a bigger issue is that these are thermophilic species. What that means is that they are better acclimated to warmer temperatures. If we look at niederhauseri, the optimal growth temperature is around 86 degrees Fahrenheit. The other 2 species are sitting at about 90 degrees Fahrenheit. But there are some isolates out there that have been shown to be able to grow up to 99 degrees Fahrenheit. And continue discussing why we see this emergence and continuing issue of Phytophthora disease. While as more pistachios are planted, right, there's going to be, most of these new plants could be going into this marginal soil that are heavy hard pan soils that are high in saline and saline sodic soils. And what this means is there's poor water infiltration. So you're getting that standing water at that soil level that is going to be benefiting Phytophthora diseases. Other factors include over-irrigation from that drip line being too close, and of course the use of surface water that could be introducing those Phytophthora propagules into an orchard site, as well as just warming temperatures and also the effects of the atmospheric rivers causing these flooding conditions, especially what we were seeing back in 2023. And right, when we think of disease management, as I mentioned, these are water molds, so it's going to be looking at how the first step is going to be looking at your irrigation. When we go out to a farm site that has pistachio issues, we always go and look at that drip irrigation. As I mentioned, having those emitters, especially right above the crown and roots, is going to benefit Phytophthora diseases, and so we always recommend to kind of move those emitters away from the root zone or go ahead and offset them so that they're not directly over the crown. And as I mentioned, basis for Phytophthora management is going to be managing your water, avoiding long soil water irrigation sets, right? We usually don't recommend longer than 24 hours in order to avoid that puddling around the root crown. Not only that, but improving water penetration or planting on berms. And what the research that our lab has been looking into over the past 6 years, I think now, is looking at rootstock selection and fungicides available for best disease management. And so to talk a little bit about the work that we did for our rootstock selection, I won't go through and showcase everything, but kind of give the quick highlights of what we did. Right, in pistachios, there's 3 rootstocks available. Those are platinum clonal plants, UCB1 clonal or seedlings, and PG1 seedling plants. And so our first objective for management strategies was determining the relative tolerance of clonal UCB1, PG1, and clonal platinum rootstocks to Phytophthora crown and root rot. The way that we assessed their susceptibility was using 2 methodologies. One was the stem inoculations, and the other was root inoculations. Very quickly, for these stem inoculations, we'll go ahead and wound the pistachio plant, and then go ahead and put a mycelial plug with the various Phytophthora species, allow them to incubate, and then measure the lesion length afterwards. After which, what this graph showcases here is that UCB1 overall would have these larger lesion lengths with respect to PG1 rootstocks and platinum clonal rootstocks. And so this kind of began our hypothesis that platinum rootstocks are more tolerant to Phytophthora crown and root rot infections compared to these other 2 rootstocks. And in order to simulate an infection cycle that's more natural to what we see in the field sites, we also did inoculations affecting the roots using these Phytophthora propagules, these swimming spores, these zoospores, and then assessing the plants for plant death, crown rot lesion, and as well as percent loss of root mass. And once again, I kind of won't go through all the data we've collected, but share the take-home message. This is one experiment where we showed that no platinum rootstocks infected developed crown rot symptoms, whereas with PG1, you had about 60% of the plants that developed crown rot symptoms, and those UCB1 clonal plants that we tested developed 100% crown rot infections. And so in the conclusion of our rootstock selection experiments is that, platinum is the rootstock of choice for managing Phytophthora crown and root rot infections in California, especially if you're in an area that is more prone to standing water, or if you already have infection already in your field, and replacing infected trees with platinum will give a better chance for those trees to survive and thrive. Now there is still some issues regarding the performance of platinum against verticillium, and we could chat about that a little bit more if you find me afterwards. And so switching gears, right, the other tool at our disposal for managing this disease is, of course, Mycota fungicides. And we can't talk about fungicides without talking about resistance. Classic fungicides that are used include various phosphonate products and mefenoxam products. And there has been reports in other agricultural systems and other Phytophthora species, not those present in pistachio, that due to overuse have developed some form of tolerance and resistance. And so when we're beginning to look at our fungicides to use, right? We really want to expand the FRAC groups and expand those that are available to be able to be registered for pistachio use. Those are highlighted in green. You have products like mandipropamid, oxythiopiperlin, fluopicolide, ethoboxam, all of which belong to various different FRAC groups numbers. And the overall goal that we wanted to evaluate is to see how effective they are in controlling this disease. And if we were able to show that all of them are highly effective, then of course that allows us to build these rotational spray programs from fungicide resistance, developing to any single one of these products, and just kind of jumping ahead and thinking ahead from preventing a resistance from even happening. And right, so that's kind of some of the objectives. What we seek to assess was first looking at baseline sensitivity assays for the 3 Phytophthora species, as well as evaluating the fungicide efficacies in these short-term greenhouse trials, also looking at the systemic activity for these different products. And then we also did a couple of experiments looking at the number and method of application for potassium phosphite, because it is the only product available that can be applied as a foliar or as a chemigant. And so for objective 1, looking at these baseline sensitivities, I won't go through the exact process, but we use 2 methodologies to assess the EC50. And all that means is the concentration of a chemical compound for which we saw 50% mycelial growth inhibition inhibited relative to the water control. And so for phosphites, we use serial dilutions. And for the other products, we use the spiral plating method that was kind of pioneered by Jim Matascavage and their lab down at UC Riverside. And so this is just a quick table showcasing those baseline sensitivities, right? The purpose of assessing these sensitivities to begin with is just to have the tools to be able to monitor whether these Phytophthora species populations over time have a shift in their sensitivity, and to allow us to monitor for resistance to any potential fungicide product. And I do want to point out that out of those tested, oxythiopiperlin was shown to be more toxic, 10 to 1,000 times more toxic than the other products out there. But aside from baseline sensitivities, we kind of wanted to test this in a more natural environment. And so we did conduct several greenhouse trials testing the efficacy of these various products with infected plants. And so the way that we did this was inoculating small plantlets in greenhouse with this infested oat and vermiculite mixture with Phytophthora niederhauseri. We added that around the crown area. As I mentioned, that's usually where we see infections occurring, and so we kind of wanted to mimic what was being seen in the natural environment. A week later, we'll kind of go through and apply the various fungicides and let them incubate for about 17 weeks. And then afterwards, we would assess the efficacy of these products by counting the propagule counts in the soil. And all that means is counting the population of Phytophthora that's kind of in the soil area, and as well as looking at how effective they were at reducing the amount of root rot infections. And so that's kind of what this first slide shows, right? We're looking at those propagule counts in the soil. And so this took a lot of time talking about the grad student that's going through and counting propagule soils and single plates. But what our results showed here is, right, when it comes to knocking down those propagules and populations counts, products like oxythiopropylene at either the high or low concentration was highly effective. We weren't able to recover any propagule counts in this experiment, but nonetheless, the other products like potassium phosphate, ethoboxam, mandipropamid, fluopicolide, and mefenoxam were all still highly effective at reducing those propagule counts relative to that non-treated control. And right, the other part that we wanted to assess was looking at the reduction in Phytophthora root rot infection after these fungicide treatments. What we saw once again is that all products were highly effective at reducing root rot infections, with oxythiopropylene and mefenoxam showing that they were the most effective products. But nonetheless, the other thing that we wanted to evaluate was looking at the capability for these products to be systemic. And the reason why is, as I alluded at the very beginning, the crown rot infections form at that soil line. So in order to provide good systemic protection, that fungicide would need to be carried at least into that 5-inch margin of where those infections begin. And so the way that we evaluated this was using field-planted plants. We did a soil drench application, and then following that soil drench application of the various products, we'll go through with the mycelial plug, and then treat the plants 3 times over the course of 3 months. And afterwards, we evaluated systemic activity by the resulting lesion lengths. And so when field inoculations were done roughly 28 inches above the soil line, we really saw that potassium phosphate and mefenoxam were the only products that were moving up that high and showed the most systemic activity. We once again repeated these experiments at that 3-inch inoculation levels to assess whether or not these other products were still able to move into that area of high susceptibility of infection from Phytophthora. And what we saw here was that all products were, to some extent, being taken up into that crown rot zone and have the potential to provide some form of protection from infection from Phytophthora. And our last objective, as I mentioned, the phosphates are the only ones that could be applied as foliar and chemigation, and so we wanted to kind of look into whether or not the number or mode of application has some kind of effect in reducing those canker lesions. And our experiments were set up in a borrowed orchard, so we were not inoculating the trunks because we did not want to kill anybody's trees. So we had to do these inoculations up in the branches. But then, of course, after these inoculations, 1 week later, we would then come in as a chemigation or as a foliar application of phosphates, and then repeating these applications 1 or 2 or 3 times, depending on what the treatment was. And then, of course, looking at those lesion lengths in order to measure the efficacy of the various treatments. So our first experiment conducted in 2023, what it really showed was that those foliar applications were highly effective at reducing the canker lesions relative to the control. And there wasn't really any difference seen between 3 applications or 2 applications. If we look at the drip, the drip line treatments, the chemigation treatments, there was also no difference in canker lesion when comparing 3 versus 2 treatments. And last year's data, we were comparing 1 versus 2 applications. And what we showed here again was that the foliar application applied twice was the most effective treatment when compared to just a single foliar treatment or a single drip treatment. But the kind of grain of sand here is that we might have seen more efficacy with foliar treatments because we did have to do those. We did have to use branch inoculations, and so maybe that product was being more concentrated in the branches given the foliar treatments. But nonetheless, the kind of take-home message of of our fungicide work is that all products tested were highly effective at reducing and controlling Phytophthora crown and root rot infections. So all of them are going to be highly valuable for implementing and providing as an extra tool that farmers can use for managing this very serious disease. Right, as I mentioned, all compounds tested showed to be systemic and being moved up into that susceptible area at that soil line. And of course, when it kind of comes to these foliar applications of potassium phosphates or chemigation applications of potassium phosphates, all were shown to have the capability of reducing those canker lengths regardless of the form in which they were applied. And so now, if we look back at the table I had shown earlier, when we look at disease management, our research has been helping to fill in the rootstock selection, right? We would recommend that farmers use platinum rootstock, especially if they have a Phytophthora disease issue. And when it comes to fungicide applications, our research has showed that the products mefenoxam, potassium phosphate, and oxythiopurpulin, which at this time are the only 3 products registered for pistachio, all of them are highly effective at managing this disease. And the general comment that we give is that, soil applications and chemigation applications should be made in the spring when you have those root flushes going on, and increases the chance of those products to be taken up into the crown area. And then, of course, foliar applications in the spring with the phosphites. And of course, like many of fungicide products, they're best used as a preventative measure rather than a curative measure. And so when it comes to future perspectives and what we seek to be continue doing in the lab is that we're working with the various company reps to register the new products like ethoboxam and fluopicolide in order to increase the tools available for managing this disease. We're doing continuous work working with the pistachio breeder Pat Brown out of UC Davis to continue evaluating new pistachio rootstocks that have an even higher tolerance to Phytophthora. And then I do want to also emphasize that, as I mentioned, the atmospheric rivers coming out of 2023, we saw huge amounts of flooding from nearby rivers, from nearby levees, massive amounts of water that were being moved around and introduced into these pistachio orchards. And from this movement of water, we have been detecting new Phytophthora species affecting pistachios. That's Phytophthora nicotiana and Phytophthora crassumora. And so our lab seeks to continue to evaluate the threat that these new pistachios, or Phytophthora species, will have for pistachio plants. And so with that, I want to thank my lab members because it is a lot of work to process these roots and do these fungicide trials, and so couldn't do it without them. Of course, the California Pistachio Research Board for funding this research, and of course, Duarte Trees and Vines for donating the plants used in our fungicide trials. And with that, thank you for your time, and I'll take any questions.